So hello dear friends, today we will talk about protein digestion and absorption, and this is the very important topic in gastrointestinal block in your medical and paramedical sciences. So basically we will talk about the physiological part, and this however this will be equally effective in the biochemistry block also, and biochemistry also, and simultaneously means I will talk about the physiology and biochemistry simultaneously. So for detailed lectures, I hope you will go through the detailed lecture, uh, and for further study, uh, you can refer to the textbook of the medical physiology by Guyton and Hall. Uh, in the same way, like Ganong's physiology, other references are like Lippincott's illustrated biochemistry, then D.M. Vasudevan biochemistry. So these are the sources. You can refer these sources for further uh, knowledge. So, protein digestion. First, we should know that where protein digestion starts. So, as I mentioned in earlier lecture in the carbohydrate digestion and absorption, the protein digestion starts from the stomach. Stomach starts here. I mean in stomach, but the carbohydrate digestion I have mentioned in detail in the carbohydrate digestion and absorption lecture physiology. You can go through uh, that lecture too. So the MCQ and viva question will be like carbohydrate digestion starts in the mouth and continues uh, in the stomach, and final digestion occur in the small intestine, upper part of the small intestine, like mainly the duodenum and jejunum. Then when protein digestion, when we talk about protein digestion, it starts in the stomach. It starts in the stomach. So let's talk about the protein digestion in the stomach. So before entering into the uh, detail, I will mention some terminology here. Please remember, like this, whenever I talk, I mention uh, proteases. Proteases. This is the protein digestive enzymes. Protein digestive enzymes, and when I talk about the zymogenes, zymogenes. So this zymogenes means inactive form of the enzyme. It represent inactive form of enzymes. Enzyme is the zymogen, or you may call it as the proenzyme. 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 Then yes. Okay, other terms I will mention whenever they come. So stomach. So in stomach, first I would like to clear that in stomach the digestion occurs in the gastric medium. In small intestine the digestion occurs in the basic medium. Like in stomach the favorable range for the enzyme action is the 2 to 3, 2 to 3, and um, that enzyme action can occur till pH 5 or below. pH below 5, or some authorities also believe this enzyme will work only pH below 4. So moral of the story is that in stomach digestion occur in the acidic medium. Acidic medium. Why I have talked in previous lecture detail that enzyme action, enzyme is a specific for work in a specific temperature and pH range. So that is the reason, that is the reason. And in small intestine, the in small intestine the enzyme action will occur in the slightly basic pH, near around pH 8. So I will not mention again and again. So here is the proenzymes. Then the digestion, for the digestion of this protein, there should be secretion of the enzymes. Then enzymes in the, so in the stomach, let's suppose this is the stomach, and this is continuation of the small intestine. Here is the sphincter, this is the cardiac sphincter, and this is the pyloric sphincter. You know, in anatomy it is very important, like junction between this esophagus and stomach, duodenum, esophagus and stomach is a cardiac sphincter. Junction between stomach and duodenum is the pyloric sphincter. So how to remember? Remember, like in ABCD in alphabetical order, C comes first, then comes P. So it will be it will be in upper opening, and this will be in lower opening. So forget about that. What I was trying to mention, that in stomach there are the gastric glands. Gastric glands, it contains two types of the cells, mainly two types of the cells. There is the something called as the parietal or actinic cells, parietal or actinic cells, and it will be secreting hydrochloric acid and Castle's intrinsic factor, which is required for the absorption of the vitamin B12. I will talk in detail in the vitamin lecture. So, and this will be your again your MCQ and Viva question. Which vitamin is for absorption of which vitamin Castle's intrinsic factor is required. So this will be secreted by the parietal cells, and there will be something called as the chief or peptic cells. It will be secreting the pepsinogen plus prorenin. That is the zymogenes or inactive form of the enzymes. So first let's talk about that enzyme which is required for the protein digestion. Their main enzyme is the pepsinogen, and next is the prorenin. So these both will be converted into active form, that is pepsin and the renin. Remember, this renin is not the renin. This renin and this renin is a different double N. Here is a double N, here is a single N. Different, but students, what they think that this renin is the enzyme. This renin is the enzyme, and this renin is the hormone. So they differentiate like that. But dear friend, this renin and this renin both are the enzymes. For this there is no doubt, this is the enzyme. This it is functionally the enzyme, because it is converting the angiotensinogen protein into the angiotensin 1 peptides into the peptides. So this is causing breakdown of the protein molecule. So this is functionally, this is the enzyme. However, this renin may show some hormones like characteristics, like it is secreted in the kidney, by juxtaglomerular cells, and it is transported through the blood stream, which is the different for the enzymes. That enzyme is transported through the duct and secreted by the ducted gland, that is exocrine gland, and that is secreted from the endocrine gland, and it act on the distant work like secreted in the kidney, and it act in the uh, liver, So by characteristics, this may show some characters like hormone, but actually functionally this is functionally this is also enzyme. So uh, sorry for the deviation. This enzyme will be causing the digestion of the protein in the stomach. So, and for this activation of this enzymes, for activation of this enzyme, the hydrochloric acid will be needed. Hydrochloric, again MCQ and Viva question will be like which factor causes the activation of these enzymes in the stomach. So please remember hydrochloric acid. Why acidic medium is needed? You are seeing here. First for the activation, then for the enzyme action, the acidic medium is needed. So, and also remember, please also remember that this pepsinogen once gets activated into the pepsin, this pepsin can activate this pepsinogen itself too. But when asked in MCQ and Viva, your answer should be this hydrochloric acid, that is activator of this pepsinogen and prorenin. Now move on to the digestion. So protein in the stomach, proteins will be break down into the by this enzyme pepsin proteoase proteoase. Here is a protease, do not get confused. Age for the enzyme, this is the proteoase. Proteoase peptones and peptides. Now dear friend, please um, don't get confused that this all are the protein breakdown product, and why different name my dear. First on the basis of the amino acid, how much amino acid containing they are, and then next is their solubility or on the basis of the size. These are the different, like first on the basis of the size, this proteases is the larger, then the propeptones, and then the peptides. Again peptides and proteins, you may also get confused. So I would like to mention, clear your doubt that according to the some many authentic books like Harper's Illustrated Biochemistry, the peptides are the lesser than 50, sorry, up to 50 amino acid chain containing peptides are the peptides, and greater or equal to 50 amino acid chains containing proteins are called the proteins. So remember these two. These are the breakdown product of the proteins in the stomach. So pH range is, I have previously also mentioned, pH range is the 2 to 4 favorable pH, sorry, 2 to 3 favorable pH for this enzyme action. Then there is the now renin. So what is its function? Renin actually causes the digestion of the milk protein milk protein like casein, milk protein casein, renin, in the presence of calcium ions, it will cause the calcium paracasinate, change the this casein into the calcium paracasinate, and the further digestion will be caused by this pepsin. Further digestion of this product will be caused by the pepsin, like uh, peptides and others. So remember here, this process, this process is known as the, this process up to here this process is known as the, like, curdling of milk, curdling of milk, curdling of milk. So question will be, your question will be in MCQ and viva, that curdling of milk is the chemical change or the physical change, like here is the milk changing into the curd. So this is the chemical change, you can see here, as chemical change is occurring, calcium is binding and form structure is changing, this is the chemical changing. And again the final digestion will be carried out by this pepsin and other subsequent enzymes. Now, so this renin is mainly actively secreted in the in large amount secreted in the like infants up to the children, infants and children, and as the people ages, as the person ages, this secretion of this renin will be decreased, hence the milk digestion is reduced in the adult and older people. Now again question, like some students may get confused, I don't think that may have that may be the case there, but like what students think, that lactose is the milk from derived from the milk, and it is digested by the lactase, that is carbohydrate, and milk also contains fats, and fat will be digested by the lipases. So please remember, milk contains both sorry, all three: carbohydrates, that is lactase, digested by the lactase enzyme; casein, a type of protein, digested by this renin and ultimately by pepsin; and the fats in the milk will be digested by the lipase. So remember these, please do not get confused. Now move on to the digestion of protein in the small intestine. Number 2 is the small intestine, digestion in small intestine. That is, mainly digestion part is the duodenum and jejunum, duodenum and jejunum. The ileum is mainly for the absorption, jejunum, and in ileum there may occur the digestion also, if not completed in upper part. So that is not being issue. So in small intestine, I previously mentioned that the this requires the basic medium, enzyme action require basic medium, and there are several hormones that causes the secretion of the pancreatic enzymes, like the major secretion, like main activator is the cholecystokinin hormone, and some amount of the secretion also can be stimulated by the secretin. So major stimuli is the cholecystokinin, remember that too. and GI hormones I have already talked in the previous lectures, so you can watch there. In small intestine, mainly the enzyme is the pancreatic, then there will be the uh, enzymes from the cell lining of the small intestine, that is enterocytes. So first talk about the pancreatic digestion. So from the pancreas, then exocrine pancreas will secrete the enzymes, in inactive form like uh, trypsinogen. First talk about the trypsinogen, trypsinogen. Then there is the chymo, chymotrypsinogen. There is also the carboxy, polypeptidase, carboxy polypeptidase, and there is also the elastase. This is the pro carboxy polypeptidase, chymotrypsinogen, and proelastase. These all are the inactive form of the enzymes, that is zymogens. So again these are secreting in the inactive form. First it should get activated, then it will only act on the substrate, protein substrate. So trypsinogen is converted into trypsin, very important MCQ and viva question. Trypsin by the enzyme known as the enteropeptidase. And this is also known as the, previously it was called as enterokinase, and for secretion of this enzyme, this enzyme is from the enterocyte of the small intestine, enterocytes. Source and the activation is causing by this enteropeptidase or enterokinase. This is very important MCQ and viva question, that trypsin is activated by which enzyme. So remember this. Now once this trypsin gets activated, this will cause activation of all other pancreatic enzymes, that is chymotrypsinogen into the chymotrypsin, this procarboxy peptidase into the carboxy peptidase, carboxy polypeptidase, and proelastase into the elastase. This function is carried out by this trypsin. Now my dear, again same as like the pepsin, here also this trypsin once activated can cause the activation of the trypsinogen self. But when asked in the MCQ and viva, please answer this enzyme. The activating factor for this chymotrypsinogen is the enteropeptidase or enterokinase. So this is clear from here. Now talk about the enzyme action. Now from the stomach, food is coming into the duodenum. This is the acidic content. First it should be pH will be neutralized by pancreatic and biliary bicarbonate secretion and other buffer ion secretion. This will first neutralize the pH, then this enzyme will act. Now, so what I am trying to show is there that this trypsin, how they will cause the inflammation, like from the stomach. Yes, from the stomach what is coming, like some undigested proteins may also come in direct into the small intestines, or or the this sorry digested product of the protein from the stomach, that is proteogens, peptones, and peptides peptides peptides. So what will happen, these these enzyme these enzyme act like the chymotrypsin, chymotrypsin, then there is the carboxy polypepti polypeptidase polypeptidase. All these will further digest this protein products and proteins, and then product can be like, if there are the proteins, this may repeat the proteogens, peptones, peptones, peptides, 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 and fewer amino acids, fewer amino acids, fewer free amino acids. Okay, amino acids will be very few, so I will not mention here. Mainly the peptides, main digestive product will be the peptides. So peptides. Now on these peptides, the enzymes like peptidases, different peptidases will cause will digest this product into the uh, peptides, again peptides, smaller peptides, smaller smaller peptides plus few free amino acids in the small intestine. And these peptidases are mainly secreted in the from the enterocytes, brush border uh, lining of the enterocytes, that it may secrete in the small intestine, but these peptidases act in the mainly act in the enterocytes. Once dipeptides and tripeptides like mainly here will be the dipeptides and tripeptides that will be absorbed. Both I will mention that amino acids, not only the free amino acids, but dipeptides and tripeptides are also being transported by this small intestine, unlike the monosaccharides, that for only the monosaccharide form are the transported in the uh, carbohydrate digestion. So now what I am trying to show is that why different types of the enzymes, proteins. If it is a protein, there is may this one or two enzyme should be digested, should digest these all proteins, but why there are many enzymes, protein digesting enzymes? Because I will rub here again. I will go to the some basic part, like these enzymes has the specificity, something called as a specificity, like it will break the bond at the specific site, at the specific amino acid bond, between a specific amino acid bond. So for example this trypsin this trypsin will cause breakdown of the bond of the peptide bond if only if they are if the bond carboxyl end is provided by the provided by the uh, lysine and lysine and uh, lysine lysine and arginine, yes, lysine and arginine. If the carboxyl group is provided by the lysine and arginine amino acid, then only this trypsin will act on that peptide bond. So for their specificity on the basis of the amino acid, they require many types of the enzymes. Now again the question will be, again the in the digestion there is something called as the exopeptidase and the endopeptidase. Exo and endopeptidase. Exo and endopeptidase. So this exopeptidase, first talk about the exopeptidase. Like it will contain the aminopeptidase. So aminopeptidase, what is its function? Why called so? Let's suppose this is the chain of the smaller peptide, and here I am showing the peptide bond, how is peptide bond is forming. So if aminopeptidase, and also I would like to mention here that let's suppose there are many amino acids in between, but there is the something at one side there is the amine group, and at other side there is the carboxyl group. So if um, so this enzyme will act only uh, on the amine terminal, from the amino terminal of the peptide bond. And exopeptidase, why called exopeptidase? Because this enzyme will break the peptide bond from the side of the chain or terminus of the chain only, so called the exopeptidase. And if causing from the amine side bond break, so this is the aminopeptidase. And there is something called as the carboxypeptidase. This carboxypeptidase, as the name suggests, if if it is breaking the bond at the carboxyl terminus or carboxyl end, so it will be carboxypeptidase. Now there are other many various exopeptidases like dipeptidyl dipeptidase, dipeptidase, it will break the bond, break the peptide bond like this. Let's suppose again this is the chain of the amino acid. So dipeptidase, it will break here, like it will separate from the terminus two amino acids. It will break two amino acids. And there is the again dipeptidase. So dipeptidase and tripeptidase, something called as tripeptidase. So this dipeptidase, let's suppose there is the dipeptide. So if this is breaking this bond and releasing the free amino acids, this is called known as the dipeptidase. And something there is the tripeptidase. So this tripeptidase, what it will do, it will cause breakdown of the peptide bond in the from the tripeptide. And there is something called as the omega peptidase. This omega peptidase again it will uh, break the peptide bond. Omega peptidase like two amino acids dipeptides, it will break into the from the terminus like dipeptides same as like this dipeptidase, and there are other peptidases also. So these are the exopeptidases, and this exopeptidase this peptidases mainly present in the enterocytes, and there if there is the absorption of the dipeptides and tripeptides, that will break down into the free amino acids, and in the small intestine lumen of the small intestine also. So small peptides will be digested like this and release free amino acids, and something so there is exopeptides, so there must be the endopeptidase, so also endopeptidase. Endopeptidase, so besides these enzymes, all are the endopeptidase like this trypsin, chymotrypsin, carboxylic peptidase, elastase, these all and pepsin also, these all are the endopeptidase. So what is the difference between exo and endopeptidase? So for endopeptidase I will mention here like this endopeptidase, as the name suggests, this will break the break the peptide bond, um, not from the terminus but in the inside the peptide bond. It will break like the here in the very larger proteins. If breaking from the terminus, that is exopeptidase. If breaking in between or from the inside, that is the endopeptidase. In examples are the these all are the endopeptidase. So this is the difference between the exopeptidase and endopeptidase. Now moral of the story is that in the lumen uh, all the like all the proteins are not digested into the free amino acids. The main observable form is the dipeptides, tripeptides, and the fewer amino acids. So let's talk about the absorption, and at that absorption okay, move on to the absorption. So absorption of amino acids, free amino acids, dipeptides and tripeptides, these all are the absorbable form of the protein digestion, tripeptides. So tripeptides, so digestion mainly occur in the lower part of the small intestine, that is the ileum. So I will draw here some cells like let us suppose this is the lumen of the small intestine, that is ileum, lumen, and this is the paracellular or interstitial space, paracellular or interstitial space, interstitial space. So digestion how will occur? Sorry, absorption how will occur? Same as the monosaccharide like glucose and galactose transport it will be transported through the carrier protein called as the sodium amino acid co-transporter. Sodium amino acid co-transporter co-transporter transporter transporter this will be the transporting like free amino acids, uh, dipeptides and tripeptides tripeptides along with the sodium ion sodium ion sodium ion. So for this digestion, and here on the basolateral membrane, you know, there is the sodium potassium ATPase or sodium potassium pump. So this is causing 2 potassium in and 3 sodium out, 3 sodium out. Same on lateral and basal membrane, there will be also the 3 sodium out, and 2 potassium in, So these sodium potassium ATPase or sodium potassium pump is causing the net deficiency of the sodium ions inside the cell, and this will drive this sodium this is the sodium down the electrochemical gradient, and this is against the electrochemical gradient. So this is the primary active transport, and so this will be the secondary active transport mechanism, like sodium like electrochemical gradient created by this primary active transport mechanism is used by this transport. So this is the secondary active transport mechanisms. By this mechanism, this amino acids, dipeptides and tripeptides are being transported, but when it will be finally carried into the through the interstitium in the venous portal system, hepatic portal system, through the small intestine, blood vessels, these dipeptides and tripeptides must be digested into the must be converted into the free amino acid inside the enterocytes. So here will be the only amino acids, and these amino acids will enter into the blood, and dipeptidase and tripeptidase, these enzymes in the enterocytes will be causing the digestion of these dipeptides and tripeptides. So remember, what you should remember is that in the blood, the only observe in circulatory form of the protein digestion is the amino acid. Now if these dipeptides or other longer peptides gets absorbed in the small intestine and move into the blood, that will cause the food allergy. So remember this absorption. Now this again this absorption, but different other books also suggest, other authorities also suggest that there are different specific transporter for this dipeptides and tripeptides beside this sodium amino acid co-transporter, and that will be like proton linked peptide transporter cause the digestion absorption of this dipeptides and tripeptides. So remember that too, and that by that mechanism that dipeptides and tripeptides can also be reabsorbed, and for this reabsorption there according to the specificity, there is the many types of the this type of the transporters, like medical physiology guidelines and all suggest there is at least 10 type of the transporter according to the specificity of the enzymes, and some book like lipin codes by illustrated biochemistry says there are the at least 7 identified different types of the transporter. So moral of the story is that there are different types of the transporters for the different amino acids. So this is all about the absorption of the protein. Hope you enjoyed the lecture. Thank you for watching. By application of this mechanism, what is its clinical application? Let's see. There is something called as the super ORS. What is it? The ORS fortified with amino acid is called as the super ORS. Super ORS, what is its function? By using this mechanism, knowledge of the physiology, the ORS formulated as super ORS, and it is very effective in the rapid retention of the electrolytes and fluids in case of the diarrhea, and in case of the diarrhea or other fluid and electrolyte imbalance cases, like this sodium, this amino acid of this ORS will be causing the absorption of the sodium, and if sodium is absorbed, then to maintain the osmolarity through the paracellular pathway, there will be the absorption of the water also. So fluid and electrolyte and other types of the electrolytes also will be absorbed, and this will rapidly replenish the fluid and electrolyte loss or imbalance in case of diarrhea or other type of the imbalance. So this is the clinical application of this amino acid transport, same as like the glucose co-transporter sodium. Why glucose is added in the ORS? You know that because that glucose is causing the absorption of the sodium via the secondary active transport mechanisms. So this is the clinical application of this transport mechanism. So why I am saying time and again that if you know the physiology, you can easily understand the pharmacology and formulate the drugs to modify. What is drug? Drug is any substance or product that is used or intended to be used to modify or explore the physiological states and change the pathological states and change the pathological status. So this is the drug, basically drug.